We share 99.9% of our DNA with every other human. But the trillions of microbes living in our gut tell a much more personal story. In fact, no two microbiomes are alike, and those differences shape how we respond to food, drugs, and disease. Today's guest wants to turn that hidden biology into personalized medicine built around the patient, not the population. Welcome back to Humanify Health Tech, where we explore how AI is rewriting the rules of healthcare and putting people back at the center of where they belong. I'm Brian Chee, Senior Director of Content and Communications at NetHealth, or your host for short. Today, I'm joined by Dr. Jenny Yang, co-founder and CEO of Outpost Bio, a company combining lab-grown human microbiomes with AI to make medicine as unique as the people it's meant to treat. Before she took on the microbiome, Jenny was using AI to hunt for cancer drugs. She earned her doctorate at Oxford as a Marie Curie Fellow, one of Europe's most competitive research fellowships, and built her career at the rare intersection of real bench science and machine learning before co-founding Outpost Bio in 2025. She's that rare expert who's equally at home growing microbes as she is building AI models. And today, she's here to help us make sense of both. Good luck with that, Jenny. But thanks for joining us.
Thanks so much for having me.
Awesome. Okay, so first question I want to jump into is it's going to be a little basic, right? But I think it's good to sort of set there, begin the conversation there. We're about to spend this entire conversation on the microbiome. So before we do that, let's just say that most people have never had to think about it as a health factor at all. So, first question, what is a microbiome? And in your own estimate, how much of a person's health actually traces back to their microbiome versus their genetics?
That's a great question. So I guess first with the definition of a microbiome, it really is the community of, like you mentioned, trillions of microorganisms that inhabit a specific environment. And I guess to contextualize this conversation, we can focus on the gut. So this would include all the bacteria, viruses, or fungi that live in your gut, but there are other microbiomes. So there's a microbiome on your skin, in your mouth, etc. But it really is that community of organisms that inhabit a certain environment. And I would say a lot of people actually have thought about the microbiome, but it might have been phrased differently. You probably hear the term gut health a lot, and that's probably much more tractable. But I'm sure a lot of people think about, the foods they eat and how that affects their health, and that is directly related to the microbiome.
Awesome. So one of the things I noticed in doing some research for this podcast, I noticed that you have a lot of passion about this topic, a lot of enthusiasm for your mission at Outpost. And one of the things that stood out to me was your work with Professor Stephen Jones at the Genome Sciences Center. That seemed to be a key point in the journey that you're on with Outpost. What was it about that experience that led you to where you are today?
Yeah, I think back very fondly and owe a lot to my career journey because of Steve. So I started my undergrad as an engineering student, but wanted to have some experience in research, specifically within healthcare. And Steve gave me an opportunity to do an internship in his lab that one summer. And he really introduced me to the whole concept of both AI within the healthcare space and personalized medicine. So I got to gain experience working alongside both machine learning practitioners, bioinformaticians, and clinicians on building tools that would help push forward the field of personalized medicine. Specifically in his lab, we were focused on oncology, and we really wanted to have a better understanding of how we could both classify cancer subtypes, but also do better patient drug matching so patients are matched with the drug that would work best for them. And specifically, we were focused on human genomics. And I also joined the lab during a really exciting time. So this was probably over 10 years ago now, when we were really seeing the onset of a lot of big data, specifically in the genomics and transcriptomics space. So it was very exciting to see the growth of all of that. And I would say that really got me fully passionate about pushing forward personalized medicine. And I feel like I've been trying to pursue that at every step of my career since then.
Yeah, it's exciting to me as a mere mortal, as a human being. That what you're doing is incredibly exciting, because we all know that feeling of not feeling like a person when you go to the doctor, or when there's struggle in finding answers to your symptoms, and having that experience. So, and that's part of it, right? That's part of what's so exciting here, is that patients experience illness as individuals, yet medicine has always treated patients as a population, and the greater pool, rather than take that more personalized approach that you're putting together. Why has there been such a disconnect between how we treat patients and what medicine predicts? And follow-up question, or two-part question to that: how can microbiomes close that gap, or more broadly, what can it actually solve?
Yeah, that's such a good question. And I think it's such a big question as well. I think to start, human biology is just so complex. And when you take into consideration the individual rather than the population, you're adding a lot more complexity. And to be fair, I think there are a lot of incredible patterns to be learned. Like, I think you see one case and you can take that anecdote and all the lessons from that and apply it to a new case. And I think that's why we've really taken this pattern recognition approach to look at health on a population scale. And I think there is a lot of truth in that. And we've seen it, especially with even like some of the generics, like generic therapeutics that have come out. Like it does work on this population scale, and that's because we can find certain pathways on this causal level that relate to all of us as humans. And then technology is also growing at the same time as our knowledge of just biology. So when I got experience in the genomics, specifically the human genomic space, I was able to see how we can identify exact genetic mutations that lead to particular subtypes of cancer. And then people are now developing therapies that target that particular mutation. And that's super effective for better understanding how we can start refining the resolution that we try to diagnose people with, or cure people with. But I would say it is relatively new. Like, I think it's been a journey that people have been on in the last 20, 30, even 40 years. But as our knowledge of this complex space is growing, technology is also growing. So I think we are limited by that pace of development as well. But it's a hugely complex task. People always talk about how the human is a system that is impacted by many different things. So we're impacted by our human genomics, we're impacted by our environment, our eating habits. So there's just so many things to consider that it makes it really complex to look at everybody on a personalized level. I do feel like the microbiome space is also one of those growing spaces where now we might have the right tooling to be able to add it in as an analysis that brings us more to that personalized level. So I, yeah, because I've spent a lot of time and seen the growth in the genomic space and seen the impact that's had, I think it's time to add all the non-human DNA. Because, like you mentioned in your intro, when you consider, the entire audience and the world, we're 99.9% identical in our human DNA. But when we look at our gut microbiomes, we can be up to 90% different. There's a lot more genetic diversity in there that when we want to consider what makes us really individual, we are going to have to add in this piece of the puzzle. But it is just a piece of the whole human biology and whole human system. So it is a largely complex task, but I think it will add a lot of extra useful information to bring us one step closer to personalized medicine.
So it sounds like to me, and please educate me here, right? But it sounds like to me like it's a journey on understanding more personalized patterns, and continually working to create more personalization within the patterns that we've already been basing our medicine on all along. It's just that from 50 years ago, the patterns we were looking for were broader, and now, thanks to your work and the work of a lot of people that are dedicated to this, it's about finding more personalized, more unique patterns to be able to find cures, find solutions. Do I have that right?
Yeah, absolutely. And like adding to the patterns that we already have and like bringing them to a more higher resolution so we really get to a more personalized space.
Wow. Okay. So using human knowledge and technology to put that all together, and and actually our knowledge in the lab is greater than the ability of technology to detect, find, and build those models. And if anything is slowing it down, it's the technology. And what we see today with AI is a quantum leap, or a huge step toward being able to do that. When I'm leading up to a question, believe it or not, I really am. When did you put that together, as far as the ability and the inspiration behind that concept of lab and machine learning?
Yeah, I think there's two parts to this question. So the first part is really the scale of data that was being generated in genomics. It was during a time of incredible growth where we started with like the 100,000 genome project, and now it's probably up to millions of genomes that have been sequenced, definitely more than that. But by seeing that and seeing algorithms that could be built on it to basically crunch through a lot of data and find patterns that we wouldn't have been able to do just in our heads, that was just really incredible for me to experience. And I was also embedded in a team of clinicians. So this is the second part. So at the Genome Sciences Center, we worked directly with clinicians. As soon as someone created a new algorithm or a new tool, it was in the hands of clinicians. They were giving us feedback. So we really were building AI that was made to be translated. So seeing that effect, I think really it had a lasting impact on me. So even when I went to do my PhD later, I knew I wanted to focus on translational AI and work with the clinicians that would be directly working with the patients to implement these AI technologies. Because it really is, like you said, that interaction of knowledge and technology, and then working with the system in the way that can actually bring it in most smoothly.
Oh, let's dig a little bit into the technical side. So if we agree the medicine needs to become more personal, that's the thesis, that's the statement, right? There's technical challenge, as you've identified, to do that, right? The microbiome isn't one thing, it's trillions of things interacting in ways that are constantly changing, evolving, all sorts. I can imagine that's far too complex for myself, for mere mortals, or even traditional statistics to try to untangle that. So what can AI do today that wasn't simply possible 5 or 10 years ago?
I think we have much more sophisticated algorithms. I think we've seen incredible work being done in the actual like theoretical machine learning space and mathematical space that helps us find patterns in much more effective ways. Like before, we started with linear models, and now we have a lot of nonlinear models and a lot of other types of models that make computing these data sets much more efficient. So that's one thing. The other thing is cloud compute infrastructure is much stronger now. So again, we can take the load of data that's actually coming in to crunch through these patterns. So I think there's been both a increase in capabilities on that technical aspect. And another one is I think we're recognizing that we really need high quality data. So the techniques to generate data that's high quality and machine learning usable, these techniques are also improving. So if we can have high quality data to train these models that are now more effective and more powerful, I think we're going in the right direction to take on much more challenging problems.
Okay, so let's talk about your personal inspiration a little bit more, because I think like I listen to you talk and I hear data set, I hear algorithms, I hear things that are not necessarily personalized. You're applying this non-personal, non-individualistic approach to create medicine that is inherently personal. Is there, when you began doing this, was there a specific person or a patient, somebody that sort of really drove you here, someone that you like still picture when you're deep in your technical work? Not to be sort of Pollyanna there, that's kind of corny, but you know what I mean? Somebody that really drove your meaning here.
So, our family has been impacted by cancer, as has many families. But that actually happened later on in my career. And I would say there wasn't one person. I think the problem itself is so important. And I think my inherent passion comes from both the concept of having personalized medicine and just the innovation and kind of technical, more scientific and technical aspects of it. I think I'm fortunate enough to say that, at that time when I was trying to build my career, I our family was lucky to have not been impacted by that. And I can't, other people wouldn't be able to say the same, but I would say the mission is so important, and you can see the impact it has on healthcare and families that I was immediately drawn to it. So yeah, I think on a bigger scale, when you see the numbers of how many people are affected by this, and when you hear the statistics, like one famous one that everyone hears is 90% of clinical trials fail. But 90% of clinical trials fail today as they did 30 years ago. So why are we not moving the needle on that number? I think that just drives me to want to find new ways and new perspectives to bring into the field to hopefully move that number, because that's a lot of resources going into such an important mission that however we can contribute to making that stat better, I want to be part of that journey.
Okay, let's shift a little bit. I want to talk about open source a little bit. So there's a growing divide, right? Leaders like Dario Amodei argue that increasingly capable open models create unacceptable risks, and that he's saying basically the future may need to be closed. At the same time, science kind of has always advanced through openness, reproducibility, independent validation. Where are you at with that? You're building open models, right? What's your thought process on this? Why do you think that's the right choice for biology, and do you worry about that at all?
Yeah, so I'm very much for open source, especially in when it comes to better understanding biology. One reason I'm pro open source, especially in the microbiome field, is because it's a growing field of knowledge. And there's a lot of discovery still to be made. And I think it would be really inappropriate of us to say what we do in the lab, hidden behind closed doors, and then tell you as fact is just true. Especially in a growing field, we do need that independent validation and external validation and peer review, especially for such an important field that has implications on health and medicine. We need to make sure what we're doing is sound and proven before we, put it out in the public and have other people actually being impacted by these findings. The other thing that's really, really good about open source, specifically in the machine learning world, is I do believe in pattern recognition and the transfer of knowledge. So if we build really effective models, let's say like right now we're focused on the gut microbiome, but we have a study coming out soon that's going to demonstrate that findings from the gut microbiome can be transferred to other types of microbiomes. So we don't want people in every single microbiome field to have to start from scratch, especially if we've moved the needle forward in a generalizable way, because that sharing that knowledge with others isn't going to affect our business model or business line. Like people can work on their own tasks in their own spaces, but learn from some of the patterns that we're seeing in our particular niche. So I think people also move faster and learn faster when they're able to collaborate with others, share learnings, share findings. Another reason I am very pro open source is I've been in the machine learning field for so long that I know I've always used open source models. I've used data sets that other people have posted publicly, and I've read other people's papers to improve the projects that I worked on. And I really don't want to pull that up from behind me. I know I got here not alone. So the least I can do is give back in that way.
We're never alone. We always get to where
Yeah.
we're at, right? As a collective group, as a core team of people helping one another. But do you worry? I mean, that's Anthropic talking about that, right? Do you worry that AI is going to become too closed and that all the progress you're making is going to slow down?
I think there's certain types of AI where, in certain fields, where I understand why people need to close it. I think I've just been very lucky to be in such a positive field when it comes to healthcare and pushing forward biology and improving health that I feel like I've always been around people with very positive views on AI. Um, I think there's a lot of good things that are open source, and I think maybe it will be application dependent. Like, for example, like models that are being built in defense. I don't know if I want everyone to have access to those, and but that seems like such a specific case that um, I can understand that. I actually feel like when it comes to more like theoretical AI and the development of new models, I do feel like that is actually going to remain quite open on a general scale. And it's once you really dig down to the specific applications that we'll start to see a divide in what's opened and what's closed. And I don't know if I'm the right person to give an opinion on every field there, but as long as I feel like we're not providing, we can only control so much of what other people do with these tools. And I guess I've always tried to um, err on the side of being more positive. But it is something we actively think about, so we will make sure that like if we're using certain types of data, we get the correct ethics approval, we anonymize it in the correct way. So we'll take steps to make sure we can do things open in as safe way as we have control over.
Yeah, because I mean, such a powerful technology, such powerful ability via the technology creates a requirement to have your own red line determined and defined based on what you're doing in the application in which it's being used. Is that right?
Yeah, absolutely. Like we definitely think about this a lot, especially when we start taking in data that's linked to patients, and um, that's when we really need to decide like that stuff we'll keep in house because that's also just a safe data practice.
Do you ever worry that this conversation, this sort of theoretical, sort of slightly real conversation is that it's going to impact your own model in terms of flattening the individual signal patterns that you're trying to develop? I mean, it's a cleaner, easier answer, right? Probably. What's the warning sign that you watch for there? When does it impact the work that you're doing, in terms of finding those individual patterns and that personalized patterns?
Um, I would say the models we build are meant to learn from individual bits of data, but then they're transformed in ways that normalize them in a more generalizable way. So we are taking steps to make sure that you can't backtrack the weights to identify individual components. As the model is used, I think from the user perspective, you can put in an individual data point to get uh, an outcome that is learning from patterns that the model has seen. So I think in that sense, we can keep it individual because we're making judgments on individual data points and learning from individual data points. But the model itself inherently is the learned patterns of a collective. And then there's certain machine learning steps and actual computational uh, kind of safeguards we can put in place to make sure it's not identifiable in a backwards fashion.
Okay. All right. Let's talk about perception. Um, Microbiome, trendy, hot, right? Lots of hype around that microbiome. Not compared to AI though, because AI is all that and probably more. And you're living in a world of the most oversold words in health tech at right now, right? I mean, you got both of them. So all you need to do is add like transformation in there, and you're going to be the trendiest person in the entire world. Um, how do you keep AI from becoming the thing that dresses up weak microbiome science? Because really, it's the science, right? All of the stuff that you've been saying, all of this really insightful information is all about the science, and AI, leveraging machine learning to be able to create even more so and even better. How do you separate that? How do you keep that from becoming the thing that hides weak science?
Yeah, I think this is a fantastic question because I do feel like AI is the hottest topic right now, and um, us as a company, like in every meeting we're in, we're making sure we don't overclaim, and we're talking about validation and reproducibility and how we can validate our findings. Um, on the not overclaiming about AI side, we're very much of the opinion that if the best model is linear regression or a decision tree, we're not going to overcomplicate it with a fancy model just to say we've used a fancy model. We do not believe in reinventing the wheel. That's not productive. And even with our first launch of a open source foundation model, Waypoint, we were very clear that this is a starting point. We didn't say it was solving the microbiome. We didn't say it's going to solve every single problem. And we told people how to use good practice to leverage these models. We said fine tune it on your own data, because especially with healthcare fields, you really need to have a fit for purpose model. So we're trying to be very, very um, just open and fair about how to use these models and not overclaim what they can do. And when it comes to the actual science part, we always said Outpost Bio functions as a lab in the loop. We have the wet lab and we have the dry lab. The wet lab can help generate high quality data, but it can also validate findings that we're getting from our in silico approaches. And that is something we really care about. We want to make sure that if we say an AI model has a certain output, we can prove it again in the lab. We also think about reproducibility and external validation outside of our own lab. So we have an academic institute as well as an external CRO that are generating buckets of data for us so we can validate the findings in our own wet lab, as well as demonstrate machine learning generalizability externally. So we are trying to take steps there just to make sure we can do what we say we are doing.
All right, let's wind back and talk a little bit about the patient that's just different. We kind of started the conversation talking about that experience, talking about what you're doing to help solve that or to open up new doorways, right? So let's dive back into that for a couple questions and then wrap up. Here's a scenario: a clinician suspects that their patient is just different. There's no way to prove it. The clinician can't put their finger on exactly what it is. The data is just not available, and it speaks in averages. So they're trying to find a solution that they don't have the data for, right? Personally, and as the CEO and co-founder of Outpost, what does that feel like to hand a clinician data that actually backs up what their instinct was already telling them?
Yeah, it feels amazing, and that's where like the translation part comes in. Um, you can do so much cool science and build really fancy models that are showing very great accuracy or patterns, but the real impact is when you actually see it having a positive effect in the real world. So it really is the best feeling, and especially in the microbiome field, it's so complex, but we've seen so many wins that have been published and talked about, whether through trials or in the literature, where people can actually identify specific biomarkers in the gut that were responsible for certain side effects or were responsible for responding better or worse to certain therapeutics. And that we really want to get there, because that is the feeling that we're going towards. Like if a clinician could just take a stool sample and then find a marker that would inform a lot, that's incredible. That's a huge achievement.
I mean, that reinvigorates their reason for being, right? That for a clinician, let alone the patient, the clinician, that reinvigorates that humanizing quality reinvigorates, goes beyond the top of their license, goes beyond what they're doing in terms of getting that enjoyment and that satisfaction of making a difference in someone's life. That's huge. What about the person though? What about the patient? I mean, that person who's never been able to get a specific a specific answer to all of their allergic reactions and they've struggled for years and years and they've just been put on standard treatments. 10 years from now, what's actually different for that patient, right? The work that you're doing now, if you fast forward 10 years, what does that look like for that one specific patient who gets an actual answer that makes a difference instead of that population guess?
Yeah, it's huge. And I think everyone wants to feel like if they go to see a clinician, that clinician can say, based on your biology, this medication or this treatment is the best fit for you. That I think that gives you a sense that there's a really diligent and responsible kind of process that someone has used to give you the best recommendation. Especially when if you're suffering from something, you don't want to feel like you have to try one drug, realize it doesn't work, and then change to another. And it's this trial and error process. That's such a long process. It's wasting time that really matters. And mentally, it's so draining. So I think having the feeling that something is right for you for specific reasons, and that can be explained to you, it makes the patient process so much better.
Okay, last question. I'm going to make you choose. Microbiomes, wet lab, right? The work you do in the lab, or your experience and expertise in machine learning. Which one drives the most passion for you? Which one gets you up in the morning and drives your greatest interest?
Oh, that is such a hard question, and it is so hard to choose. I will say my dominating expertise is the AI and the translation of the AI. I'm very much passionate about both, but I don't claim to be an expert in both. So, even the way we've built up the team, we've made sure to bring in the experts for each domain. But my background has been in AI, and I genuinely am so passionate about seeing such an incredible new technology actually being translated in a way that's extremely effective. So I think I build towards a future where we can start doing things at such high throughput because we're doing it in silico. But I do think lab in the loop is really what matters for Outpost, especially at this stage, because it's not the wet lab or the dry lab that's more important. It's really working together in this flywheel that I think will really drive discovery and change and improvements.
Awesome. Jenny, this has genuinely been one of the more mind-expanding and informative conversations we've had on this show. It's easy to forget how much health lives outside the parts of biology we've gotten used to talking about. So thank you for reminding us of that, and thank you for making it all make sense. We're going to let you go now, but before we jump into our recap, but thanks again, Jenny. Appreciate it.
Thank you so much, Brian, and thank you for the thoughtful questions. I had a great time.
Yeah, likewise. Thank you. All right, recap time. Here are the top 3 things I took away from my conversation with Jenny. Patients have been telling doctors some version of the same thing for years. This doesn't work for me the way it's supposed to work for me. Doctors were listening. They just didn't have the science yet to explain why. The microbiome is a real piece of that missing explanation. Second, getting from people like you to an actual answer for you took a computational leap and a person with the passion to bring it together. Modeling trillions of interacting microbes at the level of one person's biology just wasn't tractable until AI caught up to the problem. And finally, the harder challenge going forward is scale. Proving this works for one person is one thing. Keeping that same resolution intact across millions of people, across partners and data sets, without drifting back to averages, that's the real test. And that should do it for today's episode of Humanify Health Tech. As always, thanks for tuning in. If you're joining us on Spotify or YouTube, we'd love to hear your thoughts in the comments. And don't forget to leave us a rating while you're there. Once again, I'm Brian Chee. I'll see you next time.